anyone who has tried to develop a new skill, recover from a brain injury, or improve his mind in adulthood, the brain's plasticity has been a boon. If the slow march of time only resulted in a gradual decline in brain function, aging would be a truly unpleasant experience. Instead, we can learn new skills or develop our minds throughout adulthood and late in life. Even patients with neurodegenerative diseases like Alzheimer's disease can show the emergence of new skills and behaviors. As a matter of fact, even though we live in a culture that idolizes youth, most of us wouldn't choose to be 20 again if that meant giving up all the memories, experience, and wisdom that life has given us. Because the idea that the brain is dynamic and changeable throughout the lifespan is relatively new to neuroscience, our understanding of the implications and the potential of neuroplasticity remains quite primitive. And there are certainly no easy ways to improve our minds. Many of the brain training exercises and tools that are on the market today have not been shown to be effective scientifically yet, and just like in any new field, snake oil salesmen abound. But this area of research is one of the most fascinating and groundbreaking subjects in all of science, let alone neuroscience. How do we separate the wheat from the chaff? Is our potential to improve our minds really limitless? What are the most effective ways to train our brains? Some of the first evidence that the brain can reorganize itself is in fact fairly old. Patients who lose brain function as a result of injury or stroke can often regain some of the skills that they lost with time and rehabilitation. Ironically, the view that the brain is static in adulthood was largely influenced by the experience of adults with brain trauma. Certain functions can be lost forever as a result of injury to the brain, and recovery, if it happens, is very slow. And from what we've learned by studying both plasticity and patients with traumatic brain injuries, the brain is not universally dynamic. That is, some regions are far more changeable than others, and their malleability is directly related to the functions that they carry out. I introduced my favorite brain region in Lecture 2, and couldn't resist mentioning it again in this last lecture. The seahorse-shaped hippocampus that enables us to form long-term memories. Given its function, it should come as no surprise that it is one of the most plastic parts of our brains, and not just in humans. In the autumn, as days grow shorter and food grows scarce, new cells sprout in the brains of black-capped chickadees. These beautiful, tiny songbirds are then capable of performing memory feats that even the most celebrated mnemonist cannot hope to match. In a given year, a single bird may hide up to 10,000 snacks in as many different locations, visiting each site only once, and often remembering not only where the food was hidden, but what kind of fare is up for grabs. Tracking the relative sizes of the hippocampus, scientists have found a seasonal uptick in volume in the brains of these birds as autumn leaves begin to fall. The hippocampus is also one of the very few regions in the human brain that is not only capable of rapid rewiring, but can actually grow new neurons in adulthood. For a long time, neuroscientists thought that all the neurons that form our brains are produced in the womb, and that brain development after birth largely involved the forming of connections across the brain, the pruning of unattached cells, and various other changes. Neurons do not divide the way other cell types do, and it wasn't until the 1980s that new neurons were found to emerge in the brains of songbirds. If only we all had such bird brains, thought the experimenters. But similar neuronal generation has now been observed in mice, in rats, in monkeys, and most recently, in humans. The growth of these new brain cells is restricted to only two regions of the brain: the olfactory bulb, responsible for your sense of smell, and the dentate gyrus, which is a subregion of the hippocampus, responsible for turning short-term memories into long-term ones. What's more, the neurons that are born are fairly small and make connections with only local neurons. New neurons that project to areas further away in the brain have not been observed. This is probably a good thing. In fact, very likely a highly adaptive thing, since the migration of neurons is among the most sensitive times in brain development. We don't want to mess up the wiring in a highly complex system. It remains a bit of a mystery, however, as to why, when severed, our major nerve tracts never recover function. This is why spinal injuries are so devastating. Not all vertebrates have this problem, however. Some species of fish and frogs, with whom we share an ancestral past, can regenerate severed spinal cords and other nerves. Even some of our peripheral nerves have the capacity to regenerate and heal. So why not those in your central nervous system, our spinal cord and our brain? The answer might be in part related to the location and environment of these nerve cells. It turns out that myelin, the fatty covering that sheathes axons in the central nervous system to speed conduction of electrical signals, also inhibits axonal growth. Myelin's job is to make action potential propagation more efficient, to help neurons signal to one another faster, so that information can travel around the brain and spinal cord as quickly as possible. We don't know exactly why myelin also prevents axonal growth, but we can speculate that it might have something to do with the fact that the complexity of the mammalian brain makes it important that once established, the connections between brain regions and neurons remain fairly stable. Otherwise, we might end up with a mess of wires and a total malfunction. And sure enough, the hippocampus, being one of only 2 areas where new neurons can grow, is also often the culprit when electrical activity in the brain goes awry. The architectural design of the hippocampus, with respect to how its neurons are interconnected, enables rapid and long-lasting synaptic plasticity. This plasticity underlies our ability to form new memories quickly and efficiently, but it also makes the hippocampus prone to starting epileptic seizures. The activity that causes a seizure is strikingly similar to the mechanism that allows the encoding of long-term memories, namely long-term potentiation. The hippocampus contains several subregions, and information flows essentially in one direction. Information from your senses and other parts of the brain comes into the hippocampus through a part of the brain called the entorhinal cortex. And then these neurons are connected in what seems like a loop, with neural firing propagating largely in a spiral of connected fibers across several subregions. But one of the subregions also has axonal fibers that feed back onto the neurons in that region. We call these recurrent collaterals. These recurrent pathways help us to bind information from different senses into one coherent memory trace. When you're re-experiencing a past event by remembering its details in all their vivid glory, these recurrent collaterals are firing madly. The firing of these cells is inherently unstable, so that new connections can be formed and unformed easily as you try to learn new things. But this instability can also cause sharp bursts of firing that, when unchecked, can spread from the hippocampus across the brain, resulting in a seizure. In the healthy brain, this electrical activity is kept in check by inhibitory neurons. Neurons that release neurotransmitters in response to an action potential that prevent the downstream cell from firing its own action potential. But in patients with epilepsy, these inhibitory connections aren't functioning properly. These inhibitory neurons also seem to be involved in memory formation, albeit somewhat indirectly. Neurons in the hippocampus that represent specific aspects of a memory have a baseline firing rate that is fairly sparse. That is, they don't send action potentials very often. If you turned each pulse into a sound, it would sound like this: pop, 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 pop. But when a cell is stimulated by something, say the presence of a familiar face, it lets out a burst of action potentials that would sound like this. Brrrt. You can imagine that we have to represent many, many different aspects of a memory at the same time to get a vivid sense of re-experiencing the past. If I think back to my wedding day, a flood of memories enters my consciousness. The location, the weather, the guests, my dress, my husband's vows, and so on. The inhibitory neurons seem to be responsible for keeping the baseline firing rates low in these regions, so that those neurons that are important players in a particular memory can be heard by downstream cells. The inhibitory interneurons keep the baseline firing sparse, so that when there is a signal, that burst is what the other regions in the brain can pick up. With less noise surrounding the neurons, their signal is more likely to be picked up by these other brain regions. So memory, requiring dynamic changes in the hippocampus, and epilepsy, which happens when those changes go unchecked, are closely linked. One of the most amazing things that we humans can do is remember something, just about anything, that only happened once. And we seem to have a limitless capacity to remember these single events. Given that the brain works by wiring and rewiring neurons, it seems almost mysterious that we can remember a fact that we heard at a dinner party weeks ago without having been exposed to it multiple times. If you stop and think about how this might work at the level of the neuron, it really seems to be a quasi-miraculous skill. For example, when I was a child, I learned that bats are blind, and throughout my lifetime I've heard and used the phrase blind as a bat countless times. Then, one evening, I was at a dinner party with a chiroptologist, a scientist who studies bats. In the course of our conversation, he told me that bats are not blind. In fact, they all have some vision, and some common bat species don't use echolocation to catch prey. They have developed excellent night vision instead. So now I know that bats aren't blind, and I'll never forget that fact. With one conversation, I overwrote decades of thinking that they are blind and many, many encounters with that misperception. How is that possible? Well, that's my hippocampus doing its job and doing it well. It's not just mine that is particularly effective. 2-year-olds can show one-trial learning. And then there's those chickadees. Snails can show one-trial learning too, and they don't even have a hippocampus. But they use some of the same mechanisms, long-term potentiation and molecular changes at the synapse, to lay down information for future use. Of course, snails aren't great conversation partners, and dinner table talk wouldn't result in a permanent wiring change in their nervous system. But in our brains, the hippocampus is a powerful enough region to lay down a permanent memory trace even with only one exposure. Being a major site of long-term potentiation and having the ability to grow new neurons, the hippocampus is a factory of neuroplasticity. And it gets even better. Besides laying down new long-term memories, the hippocampus is also responsible for our ability to navigate through space. Learning the layout of your house, your neighborhood, your city, and state, that's your hippocampus's job. In fact, this is one of the reasons why the method of loci is often touted as a tool for improving your memory. The idea is to associate things that you need to remember, say your grocery list, with landmarks, like the aisles in a supermarket or the paintings in a room. That way, you walk around the room in your mind's eye and you pick up each thing that you need to remember. It's no coincidence that navigation and memory are related. We even tend to believe our memories for events are more accurate when we can remember where the event happened. When patients with Alzheimer's disease have trouble remembering what happened yesterday and finding their way home, both result from a disruption in the work of the hippocampus and its surrounding structures. You probably know a family member or a spouse or a friend whose directions you never trust. Could they improve their navigational skills with what amount to brain exercises? Well, in the year 2000, Irish neuroscientist Eleanor Maguire peeked into the brains of arguably the world's best navigators, the black cab taxi drivers in London. To become a licensed black cab driver, a person must pass a tremendously difficult test called colloquially the knowledge. Training takes about 3 to 4 years of intensive study and includes memorizing 25,000 of the streets within a 6-mile radius of Charing Cross, as well as thousands of landmarks. During training, candidates drive around London on scooters every day, learning different routes. They make an average of 12 attempts before they actually pass the test. In her first study of taxi drivers, Maguire found that the hippocampi of those who had passed the knowledge were larger than those of their civilian counterparts. But does that mean that in order to pass the test, you need to have a larger hippocampus? Or does the training phase actually make your hippocampus grow in size, just like autumn makes the hippocampi of chickadees grow? For most neuroscientists, the answer was obvious. If you have a big hippocampus, you'll be better at the task and you'll be more likely to get your license. But in 2011, Eleanor showed that the opposite was in fact true. Following a series of candidates over the course of their training, she found that they all started out with average-sized hippocampi, but the years of training led to a growth in hippocampal volume only in those drivers who managed to eventually pass the test. So successful training can result in structural changes in the hippocampus. And unless your hippocampus grows in response to your efforts, you're unlikely to pass the test. Now, the question of why some people were able to show the growth and pass and others failed remains a bit of a mystery, but the plasticity of the hippocampus is no longer in doubt. If spatial navigation training can help you grow a bigger hippocampus, what other training regimens can we engage in that are actually effective in improving our brain function and even changing the very structure of our brains? Well, just like in any other fledgling industry, the key is to become a knowledgeable consumer. What types of training have been shown to be effective by neuroscientists? One of the first studies demonstrating that training can result in permanent changes in neuronal connections came from Mike Merzenich, whose work I described in Lecture 9, and a behavioral psychologist named Bill Jenkins. Having realized that severing a nerve in a monkey's hand can cause the somatosensory cortex to remap, Merzenich wanted to see if training could achieve a similar result. Jenkins was interested in understanding the brain basis of learning. So they had the monkeys learn and practice a task that required each monkey to pay close attention to a spinning disc and apply a very specific amount of pressure to it at a particular moment in time. By applying just the right amount of pressure for 10 seconds to the disc, the monkey would be rewarded with a banana pellet. And who doesn't like banana pellets? The task was hard, and it was specific enough that it took the monkey thousands of trials to develop the fine motor discrimination required to do it well. When Merzenich and Jenkins looked at the brain maps in their trained monkeys, they found 2 results, one that they expected and another that surprised them. As expected, the map for the finger that was most important in the task had grown larger in the monkey's cortex, but only at first. As the monkey got better and more efficient at the task, so too did the neurons that were responsible for this change in performance. With expertise, the neurons worked faster and more efficiently, eventually requiring fewer cells to do the task. So training has a trajectory. First, more neurons are recruited as we learn a new skill. Over time, though, as we become more efficient, using fewer movements, our neurons reflect that difference by increasing their processing speed and assigning more specific roles to individual cells. If you think about how you learn a new sport, for example, this trajectory is clear. To swing a bat, You generally use all sorts of muscles and body parts that are unnecessary. With the right coaching, though, you can refine your movements so that you use the minimum amount of effort to create the maximum effect. We watch professional athletes like baseball players in part because they show such grace. Their movements are precise and fluid. But one of the key features of training, as Merznick and Jenkins noted, is that the monkeys had to pay close attention to the task at hand, and they had to remain motivated. Any coach or teacher knows this is true, but let's explore why. Babies, who are little learning machines, have an attention span that seems to be incredibly short. So how come they learn new things so easily? Well, it turns out that one of the features of the critical period, that time in life when learning seems to be on steroids, is related to attention. During the critical period, our brain maps are shaped and refined by experience. And if you're a toddler, everything is new, and you can't know a priori what's important and what isn't. So your brain is in a state of perpetual knowledge gathering. As your brain develops, you begin to learn what's worth paying attention to, but in the beginning, everything is interesting, at least for a few seconds. The key to this heightened learning state seems to be the brain-derived neurotrophic factor, or BDNF for short. BDNF is a protein that is active in the hippocampus and cortex, as well as a region in the front of the brain called the basal forebrain. As we've discussed, the hippocampus and parts of the cortex show plastic changes with learning. And the nucleus basalis, which is part of the basal forebrain, is one of the key structures involved in paying attention. BDNF is one of the most important proteins regulating the growth of new neurons and synaptic plasticity in the form of long-term potentiation. When neurons fire together, BDNF helps them wire together. It also promotes the growth of the myelin sheath, the fatty coating that speeds conduction of electrical signals down the axon. In infancy, during the critical period, BDNF activates the nucleus basalis and ensures that it stays on the entire time. Some neuroscientists suggest that it puts the brain into its most plastic state and leaves it on until the critical period ends. Then, when the critical period is coming to an end, BDNF shuts down the factory, ensuring stability and decreasing plasticity in the brain. So how do we put our brains back into the plastic state if we're no longer in the critical period? Well, you could turn on the nucleus basalis artificially by sticking an electrode into it and stimulating it. That's pretty invasive, but probably some lab somewhere is working on it. Or you could inject some drug that could trigger activity in the nucleus basalis to keep it turned on. There are definitely labs working on that, and such a drug would be in high demand. Movies like The Bourne Legacy, featuring people with enhanced mental powers, could prove to be prescient if we were able to develop such a drug. But for now, we need to content ourselves with more traditional fixes. We need to pay attention effortfully when we want to learn something new, and keep BDNF circulating in our system. BDNF has been shown to be released by physical activity, underscoring the importance of remaining physically active in adulthood, not only to keep the body in shape, but also to keep your brain open to learning. Just like physical exercise, if it doesn't feel like work, it's probably not working. Often in our adult lives, we reach a plateau in terms of learning new things. We get out of practice, and our brains become less plastic as a result. Some of the brain training games available on the internet or via computer are designed to keep us learning. But here too, if you plateau, simply playing a game over and over again won't have much of an effect. The best games are the ones that capture your attention and increase their difficulty as you become more proficient. Just like you'd increase the number of reps or the amount of weight you are exercising with, so too should the training software make things harder for you as you improve. And just like exercise, change is specific. If you want to improve a particular cognitive skill, like spatial navigation or attentional focus, you need to train that particular skill. The biggest problem with brain training software is that it rarely shows transfer. You might get better at the specific game, but transferring that improvement to the activities of daily living is rare. So, if you're interested in such tools, be sure to check how much of the software is based on actual neuroscientific evidence showing it to be effective. And don't underestimate the tried and true ways of maintaining a high level of cognitive functioning. Plenty of exercise and learning new skills that are complex, like, for example, a musical instrument or a language or some other difficult thing. Fancy computer games are often not as effective as something as ancient as chess. If we could implant a device, however, that puts our minds into the critical period again, would we choose to do so? How would humanity change if our ability to learn was limitless, or hundreds of times more effective than it is now? Would such a discovery blow apart the ever-widening gap between the haves and the have-nots? Or in fact, would it widen it even more because such an improvement would be only available to people that had a lot of money? We've moved away from thinking about the brain as a machine because it's hard to imagine machines that are as dynamic and changeable physically as our brains. But we've also started to use machines to interface with our brains and enhance their function. The development of brain-machine interface devices is a particularly active and exciting area of neuroscience, and in some ways, it represents the ultimate plasticity. If we could enhance our brains ourselves by adding tools to help us think, we might approach a limitless intelligence. Currently, in some extreme cases of patients with few other options, tiny computers are implanted into the brain to allow their users to communicate directly with a machine outside of the body simply by thinking about it. This truly might be the advent of ESP, or extrasensory perception. Imagine that you could move a cursor around your computer screen just by thinking about it. That's exactly the type of skill that brain-machine interfaces are designed to develop. The main application at the moment is geared towards people who have spinal cord or other injuries that have paralyzed or limited their ability to move certain body parts. For example, in 2005, the tetraplegic Matt Nagel learned to control an artificial hand by using a brain implant in his motor cortex. Just by thinking about moving his hand, he could move the artificial one, as well as a computer cursor, lights, and his TV. 3 years earlier, Jens Naumann became one of the first blind adults to buy a brain implant that partially restored his vision, allowing him to drive slowly around a parking lot almost immediately, and to navigate the New York subway system fairly well today. In these cases, neuroscientists have harnessed the brain's plasticity and enhanced it by enabling it to interact with an artificial device. I suspect that future applications of such technology will only become more common as we learn more about how our brains can change. There's still a lot of work to be done before we can enjoy all of the benefits of brain implants. For one thing, the surgery is very invasive, of course, and over time, scar tissue builds up around the device, making it less and less effective as the signal gets weaker. But the possibility is there that time will tell the extent to which we can enhance our cognition with the help of computers. As we become more and more bionic, the question of what is life that we addressed in our very first lecture resurfaces. If I switched out my arm for a machine, you'd still think of me as a live person. If I switched out my entire body from the neck down, most of you would agree I'm still me and that I'm still alive. But what if the only body part that I switched out was my brain? Now I'd be a zombie. I'd have turned into a machine. Okay, what if it was only half of my brain, or 75%, or 10%? Where do we draw the line between real life and artificial life? What if I enhanced my intelligence using only biological materials? Would I still be me? Would I still be alive? For now, these questions remain unanswered, but thinking about their implications gives us a deeper understanding of ourselves and, of course, helps us refine ever more interesting questions. The paradigm shift in neuroscience from static to plastic brains is already changing our world. In our next 2 lectures, we'll explore the remarkable medium that enables questions and other thoughts to form in our brains, electricity, and the mysterious force that makes it possible, magnetism.